Welcome everyone, my name is Tayfun Tatar and in this presentation I will walk you through the frontiers of extracellular vesicles field pioneered by HansaBioMed Life Sciences. During this presentation I will first make an introduction to what extracellular vesicles are and what our company does in this field. Then I would like to also focus on two of our bestseller product lines, namely Tangential Flow Filters and Lyophilized EVs, which are used as standards or positive controls reference materials per se in many different settings. So now let's start with an introduction to HansaBioMed Life Sciences. But before that, I would like to give some brief summary on what extracellular vesicles are. Extracellular vesicles, or shortly EVs, are cells' way of communicating with each other, and they regulate basically intercellular communication by the help of functional molecules that they carry within their cargo, such as microRNAs, other types of RNAs, or proteins. They can be classified based on many different criteria, including size, cellular origin, cargo, and cellular function, and they are available from many different sources, including biofluids, bacteria, and plants. As HansaBioMed Life Sciences, in this field of EVs, our activities can be categorized under three different categories. In the first, we do product development and commercialization in EV field, which cover purified extracellular vesicles, extracellular vesicle isolation tools, extracellular vesicle characterization kits, RNA isolation kits, antibodies, and liquid biopsy tools. On the other end of our commercial activities, we also offer custom services to our customers, which include scalable EV purification from various sources, cell media, biofluids, plants, bacteria, fungi, or even further, vesicle characterization in the form of morphology, biomarker, and functionality assays, and EV omics covering proteomics, transcriptomics, genomics, lipidomics, and metabolomics analysis, and finally EV engineering, which includes stable fluorescently labeled EV generation and surface modification. But our activities are not limited only with commercial activities, because we pursue continuously innovation through several research projects, and here you can see a short list of our currently ongoing or completed research projects. Like NAP4Dive and Innovative, which are currently ongoing EU-funded projects, or Marvel and Bow, which are recently closed, completed projects. So in order to have some overall idea on our activities, I would like to give some statistics. As for the product lines, you can see that our bestseller product lines are our purified EVs and isolation tools, for which we have basically two different audiences. For purified EVs, we basically work with nanoanalyzer developers in industrial or academic settings that need some reference material for their validation purposes or for their calibration purposes. Whereas for isolation tools, our customer audience generally includes researchers in universities or independent research institutes that require a method to isolate EVs from their starting material. And as for the sales regions, we can proudly say that we are a global company operating in all the regions that have some level of EV research, but our leading market is North America, because it is the leading market in the EV field. And since we are a European company, our European footprint is quite comparable to our North American footprint, and we are also recently expanding in Asia, even though we have a good footprint, that since the market itself in Asia is growing rapidly, we are trying to follow that trend similarly. And now, in order to give some more details about our products, especially our leading product lines, for EV isolation tools, we have a variety of solutions targeting EVs from different aspects in order to reach desired isolation depending on the application or purpose. But our bestseller product is Tangential Flow Filter, which can be used for EV concentration, purification, and size-based separation. Then we have size exclusion chromatography columns, spin columns, which can be used for EV purification from minimal sample volumes, or similarly unbound dye and antibody removal. And we also have further solutions for EV isolation. On purified EVs side, which are called or considered EV standards, EV reference materials, whatever you name it, depending on your application, we have lyophilized EVs isolated from different cell lines, human biofluids, plants, or even further based on request. And we also have a first of its kind product in the market, which we are quite proud of, which is called FLUO-EVs. FLUO-EVs are product of genetic modification done to their parent cells, which are human embryonic kidney cells, and as a result of this genetic modification, these EVs carry fluorescent proteins, namely EGFP, BFP, or mCherry, in fusion with surface markers CD9, CD63, and CD81. And by the help of this, they are stably fluorescent without an external labeling step. They do not bleach, or they don't have non-specific background signal, non-specific binding caused background signal, which make them an ideal agent for nanoanalysis measurements or calibration purposes that require a fluorescent mode of operation. For our services, the services we offer can be categorized in four. First of all, we offer purification of extracellular vesicles in a scalable way from various sources, biofluids, conditioned media, plants, or even further. And we use a method combining tangential flow filtration and size exclusion chromatography, which ensures scalability, gentle isolation, and high yield as well as purity. And of course, after isolation, we validate our EVs following MySAM guidelines. And we also offer phenotyping and imaging of EVs, including scattered and fluorescent NTA, transmission electron microscopy, ELISA, and flow cytometer analysis. We offer EV omics services covering proteomics, transcriptomics, genomics, lipidomics, and metabolomics of EVs, which can be coupled with high-level bioinformatics analysis. And finally, we also do EV engineering in the form of surface modification and stable fluorescently labeled EV generation. While doing all of these, we are always pursuing collaboration both in academia and industry, because we believe collaboration is one of the most efficient ways to carry the field onwards. And here you can see just a short list of our existing collaborators, both in academic and industrial environments. And I would like to finally mention our collaboration with our valued partner, Rocker Scientific. Rocker Scientific has been a distribution partner for us in Taiwan since 2022. And with their life science expertise, I must say that they have been very helpful for us in growing our footprint in this dynamic region in terms of EV field. Now I would like to give some details about one of our bestseller methods, tangential flow filters used for EV isolation and concentration, and I would like to start with the methodology of it. Tangential flow filtration, known also as cross-flow filtration, is based on filtrating the sample in a perpendicular direction to a laminar flow, and it is a widely used method in different biopharmaceutical or food industrial settings for purification, concentration, or difiltration of biosamples. It is relying on sub-millimeter diameter fibers made of non-binding materials such as polyether sulfone, which have certain pore size. For our case, we have options for 5 nanometer, 50 nanometer, and 200 nanometer pore size. And within these fibers, the samples are subjected to a laminar flow. And during this laminar flow, particles smaller than the pore size are filtrated out, whereas particles larger than the pore size remain inside the filter. So in its essence, it's actually a size-based ultrafiltration method. In practice, you just inject your sample within syringes to one end, and then you connect another syringe to the second end in order to push the sample back and forth until no filtrate comes out, at which point you ensure that you completed filtration. And here I would like to very briefly show you a video guide demonstrating its manual use. I would like to very briefly go over it. So here you prepare the syringe by opening the ports, and you connect the sample within a syringe to one end, and then you connect another syringe with wire lock endings. The filter is symmetrical, so you can connect in either way, and then you push your sample back and forth between these syringes. And then after you finished filtering your sample, you continue with particle washing steps and so on. So I mentioned already that it is a size-based ultrafiltration method, but what makes it different or more advantageous than the widely known and very well used dead-end filtration? In dead-end filtration, one of the challenges is that over time, particles larger than the pore size accumulate over the pores, clogging the filter through a phenomenon called cake formation. As the cake thickness increases, the flow rate decreases, decreasing the efficiency of filtration. Whereas in tangential flow filtration, the laminar flow to the perpendicular flow direction ensures a cleaning effect on top of pores, ensuring a steady filtration over time, increasing the efficiency. And by this advantage, after being introduced to the EV field, which was not originally invented for EV field, the method was embraced quite rapidly by the field, which can be seen from the exponentially growing total number of publications over the years, and it would be wise to expect even a sharper increase in the number of publications after 2024, because in the beginning of 2024, TFF was introduced as an EV concentration method in MySav guidelines, Minimal Information on Study of Extracellular Vesicles, prepared by International Society of Extracellular Vesicles, which is the rule of thumb kind of guideline in EV field. But what makes it so special that it is embraced so quickly? In order to shed light on this question now, I would like to focus on the advantages of TFF. Of course, I'm introducing a new methodology in any field as a replacement of any method. The ideal strategy would be to start with a comparison to the gold standard and EV isolation. The gold standard for EV isolation is considered to be ultracentrifugation. If we compare it with tangential flow filtration, first of all, in tangential flow filtration, there is a smooth flow of particles through hollow fibers, preventing any aggregation or any damage on EV surface, whereas in ultracentrifugation, the high force exerted on EV membranes can cause not only aggregation but also deformation of EV membranes. In tangential flow filtration, the recovery is very high, which can exceed even 80%, whereas ultracentrifugation can provide only a poor recovery of EVs, around 30 to 35%. In tangential flow filtration, you can process hundreds of milliliters of samples within minutes. In ultracentrifugation, it requires hours. It is a very time-consuming method. Tangential flow filtration can be scaled up easily from a couple of milliliters to tens of liters, whereas ultracentrifugation is not suitable for scale-up at all. In tangential flow filtration, there is a certain cutoff provided by the pore size, ensuring that below that pore size or above that pore size, depending on how you use it, there is no contamination. In ultracentrifugation, since there are many other particles in EV containing samples with closely resembling physical properties, there is sedimentation of other particles together with EVs, decreasing the purity. And finally, in tangential flow filtration, there is no requirement of any special instrument. It can be used manually, or if you want to automate it, it can be automated even with simple peristaltic pumps, whereas ultracentrifugation requires very expensive lab equipment. But of course, it is not only limited with the advantages when compared to ultracentrifugation, because there are a number of isolation methods already available in the EV field and newly introduced to EV field. So it is also wise to focus on the individual advantages of TFF. And the main advantage of TFF is actually coming from its versatility, which allows it to be used in various scenarios, including separation and isolation of small and large EVs, concentration of isolated EVs, buffer exchange, or in other words, dialysis, removal of contaminants such as unbound dyes or antibodies, EV depletion from FBS or other cell conditioning media when relevant, and elimination of unwanted proteins such as THP from urine. But the degree of exploration is not limited to the versatility of TFF, because it is a gentle method allowing for multiple different TFF cartridges to be combined in series, and you can see an example on the right. Within our internal processes, we optimized a combination of our TFF MVs with 200 nanometer pore size and TFF EVs with 50 nanometer pore size, in the way that we collect the filtrate from the first filter containing particles smaller than 200 nanometer pore size, and then we inject this filtrate to TFF EVs with 50 nanometer pore size. And finally, we collect the retentate from TFF EVs. This way, we can obtain particles sized between 50 and 200 nanometer pore size, enabling targeting specific portions of extracellular vesicles. But the degree of exploration is not also limited to coupling multiple TFFs, because it can also be coupled to other methods such as size exclusion chromatography or even anion exchange chromatography, in which case there is actually an addition of orthogonal method which increases the usability in some specific scenarios. And the main advantages TFF brings to such combinations are, first of all, concentration of EVs, because with TFF you can start with liters of samples and concentrate it down to some tens of milliliters or a couple of milliliters, which can then be input directly to chromatography columns even at lab scale, and simultaneously you can remove small solutes and large debris in a scalable and efficient way. And final and one of the most significant advantages is that it can also be automated, meaning that you can ensure high yield and rapid isolation of EVs from your starting material in an automated and controlled and standardized way, while you are making yourself busy with other things in the lab. And this is possible through the 10-FIL-100 system available from our valuable partner, Rocker Scientific. And this not only ensures a smooth isolation of your EVs, but also operates it in a standardized way. As it is always much more informative to learn with examples, now I would like to give some literature examples that use our TFF filters in action. In the first example, researchers prefer our TFF EVs with 50 nanometer pore size in order to isolate EVs from breast cancer cell lines, and their study includes shedding light on how TGF-beta facilitates cancer progression through cancer cell EVs, which is a not very well understood mechanism. And after isolating EVs, they systematically investigate their roles, and they were able to identify that TGF-beta and RAP27B takes part in cancer development not only by regulating EV release, but also by regulating the composition of EVs. In the second example, EVs are now isolated from two different mesenchymal stem cell lines, induced pluripotent stem cells and human umbilical cord mesenchymal stem cells. And the idea is to compare the immunomodulatory and regenerative potential of EVs derived from both cell lines, and this time they use our TFF Easy with 5 nanometer pore size in order to concentrate pre-isolated EVs, because they would like to introduce or inject these isolated EVs in vitro models where dosage is quite critical, and after controlling the dosage through TFF-easy, in vitro assays, they were able to show that induced pluripotent stem cell-derived EVs show indeed a higher pro-regenerative potential than their human umbilical cord-derived counterparts. In a third example, now we turn to in vivo assays rather than in vitro assays, and the starting material is bacterial cell lines, where the researchers isolate outer membrane vesicles from bacterial cell lines and concentrate it down to a desired dosage, again through using TFF-easy with 5 nanometer pore size. And by injecting these controlled dosages to in vivo models, they were able to come up with an enhanced radiolabeling method, which is quite critical for measuring biodistribution in drug development assays. In a fourth example, the starting material is now a plant source, a citrus source, with the idea that citrus EVs or other types of plant EVs offer an excellent platform for nutraceutical delivery, but their storage and preservation conditions are not optimized. So here the idea is to come up with an optimized method, and for that purpose they prefer to use our TFF-MV together with TFF-easy with 200 and 5 nanometer pore sizes respectively to isolate and concentrate EVs. So after this they test different storage and preservation conditions with an ideal or optimized method preserving the stability of plant EVs. In a fifth and last example, now we see an example where tangential flow filters can indeed be coupled with other methods. Here the idea is to shed light on the role of EVs in human limbal niche homeostasis, which is very poorly understood. And in order to perform proteomics analysis of EVs derived from limbal niche, for such downstream analysis, a gentle method is required, and the gentle method preferred is TFF in combination with size exclusion chromatography for further purification, after which proteomics analysis revealed to the researchers that EVs take part in regulation of keratinocyte development, organization of extracellular vesicle, extracellular matrix, and niche regulation. The part related to our tangential flow filters, now I would like to focus on the second bestseller product line, or the other bestseller product line, which are lyophilized EVs used in different settings for ground truth formation. But before going into the technical details, I would like to give an understanding of who needs an EV standard and why. So EV field is a dynamic field staging continuous research on different aspects. EV-based therapeutics, where EVs are considered as an ideal drug loading agent. EV-based therapeutics, where EVs form a rich source of biomarker. And also development of new methods to isolate, detect, or characterize EVs. And all such improvements or advancements require EV reference material for validation. Here you can see a video showcasing different EV-based EV characterization methods used over the years. As you can see, in the beginning there are only a number of EV characterization methods, even though they characterize EVs from different aspects. But over the years there are new methods introduced in the field, and some grow even very rapidly, such as flow cytometry with the introduction of nanoflow cytometry or nanoparticle tracking analysis, which after some time became the gold standard for size and concentration analysis of EVs. But the critical idea here is that there are new methods coming into the field every year, and all these require validation that they can indeed characterize EVs. And such requirements are met by our company by providing EVs isolated and purified from cell-conditioned media, biofluids, and plants and these are isolated and purified through a combination of tangential flow filtration and, when necessary, size exclusion chromatography or anion exchange chromatography, and they are provided in lyophilized form, which enables them to be stored up to 3 years in 4 degrees, which also simplifies the shipment quite a lot because they don't require dry ice conditions. But of course, when you tell people that you are providing purified EVs, the first question is whether they are really EVs. In order to prove that, in order to certify that, we are following MyCert 2023 guideline, which recommends EV characterization from, first of all, size and concentration aspect, which we ensure through NTA analysis, morphology aspect, which we perform with TEM imaging, and from microanalysis aspect, which we covered by ELISA analysis, including CD9, CD63, and CD81 measurement as surface markers, ALIX as an internal marker, and calnexin as a negative marker. And finally, we also ensure purity of our EVs, because it is not enough to say that your samples contain EVs, it also needs to be as pure as possible. And here our criteria is actually proposed by a widely accepted paper titled "How Pure Are Your Vesicles" from 2015, which suggests that for an EV sample to be considered pure, the ratio to the particle count within the sample to the total protein amount in terms of micrograms should exceed 10 to power 8. And this is our criterion for confirming purity of our samples. So this way you actually have some instant EVs. After mixing it with some molecular water and gently mixing it, you have your ready-to-use EVs. But how can you use these instant EVs? I would like to give some examples also to give you a better understanding. In a first example, the idea of the researchers is to develop a standardized and efficient approach for EV functionalization, which even opens new doors for EVs to be used in therapeutic settings. And for such purpose, they develop an enzymatic engineering method to the glycan regions on EV surfaces, which are available on the surface of all EVs. And for testing this method, or for calibrating this method, they used our EVs from serum and multiple cell lines, by the help of which they were able to demonstrate that their method was applicable to various EV types. In a second example, now the idea, even though it is again therapeutic, is rather on the monitoring part, because even though there are novel cancer therapeutic approaches, the monitoring of the therapy response is still challenging, and the idea of these researchers is a method to quantitate EGFR, epidermal growth factor receptor, on EVs to monitor the response to therapy. And for development of their method, they preferred EVs from HCT116 cell line, which is a colorectal cell line. And by the help of these EVs, they were able to show that quantitative measurements of EGFR can actually assist in diagnostic or therapeutic monitoring of cancer. In another example, now the idea is to measure EV distribution in vivo models, which is critical for EV therapeutics, because once you inject EVs for therapeutic purposes, you need to make sure that they reach the desired target in desired dosages. And for that purpose, they used EVs from human embryonic kidney cell lines, and by the help of our EVs, they were able to develop a qPCR-based assay through which they can measure biodistribution. And in a fourth example, now the focus is a method development, with the claim that for EV-based diagnostics, the currently available methods are not as rapid or as sensitive as required, and their solution was to come up with a light-induced acceleration of specific binding between the surface of EVs, and antibody-modified microparticles, so an immunoaffinity-based approach in its essence, where through a controlled microflow, confocal microscopy analysis in 3D, the captured EVs. For their purpose, they used two cell line EVs, HCT116 and A549, which is a lung cancer cell line. And with these EVs, they were successfully demonstrating detection of as low as 10 to power 3 EVs in 500 nanoliter samples within only 5 minutes. But I would like to also specify that development of new methods mostly come with fluorescent requirements, because in nanoscale, generally fluorescent signal generation and detection is required to be able to detect the objects. However, the traditional fluorescent labeling method comes with many issues such as stability, bleaching, or non-specific background signal formation. And the solution to this problem is brought to the field by our company as FLUO-EVs. FLUO-EVs are the product of genetic modification done to their parent cells, human embryonic kidney cells, by the help of which these FLUO-EVs carry EGFP, BFP, or mCherry in fusion with CD9, CD63, or CD81, which are common markers of extracellular vesicles. By the help of this modification, these EVs do not require external labeling, they do not bleach, they do not give background signal, and they form an ideal reference material for such fluorescent method development or fluorescent assay developments. But how can they be used? So here you can see some examples showing how versatile they are. Agnostic of the methods being used, just focusing on fluorescent requirement basically. On the left you can see it being used as a reference material to demonstrate the imaging capability in simultaneous multicolor mode in the single molecule localization microscopy approach by Abelite. And on the right you can see that it is used as a proof of concept generation material for fluorescent based detection, fluorescent detection based size distribution analysis of the Taylor dispersion analysis instrument by nanoscale matrix. Now on the left you can see a demonstration of in situ labeling and quantification of EVs in complex media by microfluidic resistive pulse sensing approach of Spectradyne, and on the right you can see it being utilized as an agent for device validation experiments in flow nanocytometry instrument by Kinetic Rivers. And finally, I would like to highlight another product line among our Lyophilized EVs, which is actually the most recent product by our company, which is brought to our customers after seeing some need in the field. So this need is coming from the use or preferred use of plasma EVs in therapeutic settings or diagnostic settings, because plasma EVs are in many schemes the most clinically relevant source of EVs for diagnostic or therapeutic approaches. However, the problem included in using plasma EVs is that more than 99% of the blood composition is formed by lipoproteins, which match quite nicely in terms of their size and density with extracellular vesicles, meaning that their isolation from extracellular vesicles or the isolation of extracellular vesicles from them with desired purity is quite challenging. And the solution is actually just using platelet EVs, another component of blood with similar diagnostic and therapeutic value or potential. But since platelet EVs do not come with such lipoprotein contamination, they ensure much higher purity, and by using this advantage as an alternative, we are offering our customers purified platelet extracellular vesicles in lyophilized or frozen form. But finally, I would like to also state that our offerings are not limited to these options that I provided in detail, because we have a very wide range of products. including size exclusion chromatography columns, centrifugation ultrafilters, spin columns, precipitation reagents, immunoaffinity isolation tools, and characterization kits. So by the help of all of these, we basically are trying to become a one-stop shop for EV researchers, where they can find all what they are looking for. And this was all I would like to say about our company and our activities. For those who are interested in, here are our references. And I would like to thank you for your attention.